Howdy, my name is Jessica Tracy. I am a doctoral candidate in Gary Kunkel's lab at Texas A&M University. During metazoan development, the regulation of specific genes at the right time is vital to proper development. One way cells accomplish this regulation is by controlling the accessibility of transcriptional machinery to gene enhancers and promoters, which depends on the location and stability of nucleosomes. Chromatin remodeling allows access to transcriptional regulators, but relies on the ATP-dependent movement of nucleosomes within the chromatin structure. Indeed, mutations within several chromatin remodeler proteins are correlated with human neurodevelopmental disorders and psychiatric disorders. One classic chromatin remodelers of particular interest to our lab is the chromodomain helicase DNA binding, or CHD8 family of proteins. I study the CHD8 subfamily 3 protein CHD8. CHD8 interacts with beta-catenin, histone H1, P53, and ZNF143, which are all factors that are important in the regulation of transcription and modulation of expression of genes implicated in crucial developmental processes. In addition, recent large-scale sequencing studies have identified loss of function mutations within CHD8 within autistic spectrum disorder patients. The number of novel CHD8 mutations observed suggests it is highly unlikely that they occur by chance, and implying that a disruption in one copy of CHD8 could contribute to increased risk of ASD. So there are 3 parts to my project. The first part of my project was to characterize the role of CHD8 in brain development. To accomplish this characterization, I performed a detailed phenotypic analysis on CHD8 knockdown zebrafish using morpholino technology and whole mount in situ hybridization. So antisense morpholinos are used that bind to the mRNA and prevent translation of the mRNA into functional protein. This will effectively drop the level of CHD8 that is present in the embryo and result in a decrease of regulation on its affected genes. Whole mount in situ hybridization was done with probes known genes that are involved in brain development, such as ODX2 and PAX2A. What I observed is that all of the markers examined so far are altered upon loss of CHD8, implying that CHD8 regulates these genes during early developmental stages. However, whether this role is direct or indirect is still under investigation. In addition to the knockdown, I am creating a knockout. So the goal is to not only recapitulate the knockdown phenotype, but also to be able to perform behavioral assays and more detailed analysis with the knockout. I have successfully created some founder lines and am in the process of screening these for germline transmission, and will hopefully have an F1 generation shortly. The second part of my project involves looking at the 2 known isoforms of CHD8. So CHD8 long contains 2 chromodomains in the ATPase helicase domain, whereas short is truncated after the first chromodomain. It is missing the helicase domain altogether. So I first performed quantitative reverse transcription PCR and discovered that while CHD8 long is expressed at a steady state level throughout development, CHD8 short actually starts at undetectable levels and increases during mid-developmental stages and drops back off. This level is also dramatically less than the full length. So now I am investigating whether these 2 isoforms function differently in regards to actual gene expression regulation by using some cell culture luciferase assays. The final part of my project will be to identify CHD8 target promoters and evaluate the overlap of the promoters of known CHD8 interacting partners, ZNF143 and beta-catenin, used in ChIP-seq. So as you can see, this project is quite detailed, and I hopefully have given you some insight into the important role of CHD8. Thank you for listening.